Today, I'm going to share with you some cool new tools that can be used for metabolic network visualization and flux balance analysis. So one of these tools is called Escher, which I talked about a little bit in our lecture last week. So Escher is a really cool tool because there are different versions that let you not only look at metabolite data, but you could look at proteomics and reaction, as well as genomic data. And a new version allows you to do flux balance analysis. So Escher works by importing COBRA models, and there are several different models available on different websites or databases. But Escher itself has three main models available just for general use. So right now I'm going to look at E. coli, which is similar to a lot of the organisms that we study in our group. And I'm really interested in looking at the central metabolism. Okay, but you could also look at other pathways as well. All right, so this is E. coli central metabolism, so all the known reactions in its glycolysis pathway, TCA cycle, and then some other important reactions over here. So I'll zoom in a little bit so you can take a closer look. So right now, for example, we have here the metabolite name, which is D-glucose. We have D-glucose extracellular, so that means it was transported out of the cell, and then it goes into the glycolysis reactions. So we can take a look at those. If you want, using Escher, you can actually add reactions that are in the known metabolic network for that organism. So you would do that by using this tool function. You can select the way that you want your reaction to go. So if I wanted to add an extra reaction to glucose, you click on it, and then I'll add its exchange reaction. So this is important for flux balance analysis, as you'll see a little bit later. Okay, so you can do a few different things with this function. But now I'm interested in looking at nucleotide metabolism. Okay, so here we go, the full nucleotide metabolic network. We have here purine metabolism, so things like adenine and hypoxanthine. Then over here we have pyrimidine synthesis and so forth. And then, of course, here we have all the enzymes involved in those reactions. So now I'm actually going to upload a different model of an organism that I've studied myself in the lab, which is Staphylococcus aureus, or Staph aureus for short, which is a pathogenic bacteria involved in many different infections. So I've downloaded this model previously, and I'm now just going to upload it. Okay, so here is my file. That is a network file, and it's a specific strain of Staph aureus. Okay, and now you have to update the names and reactions in the model. So as you can see here, a lot of these reactions that were present in E. coli are not actually present in Staph aureus, or they may have a different name or a slightly different reaction. So those are highlighted in red, so you can see which reactions are actually known for your organism and which ones are not. So now I'm interested in uploading some metabolite data that I collected from my experiment. So I'm going to click Load Metabolite Data. And as I mentioned, you can also upload gene data or reaction data. So it's a pretty cool tool for that reason. You can upload multiple types of data at the same time. So this is metabolomics data. And then our map is now updated to have the metabolites. If there are no known data for those metabolites, they'll just be in white. And then these ones here that are bigger are actually had data associated with them. So here we have hypoxanthine, for example, and adenine. Okay, and then down here we also have a few more metabolites that were in the data set, including xanthine and guanine. So you can do some cool stuff with this now. So you can go to your settings. You can change the identifiers to their names rather than their IDs, which sometimes makes it easier. You can also change your color scheme. So you might want to do a red, white, and blue type color scheme, which is what I would normally do. And then change your center of the map to a different scale. So you could have a bigger range of data shown and so forth. Okay, and that's going to slightly change the way that your metabolite data is scaled. So that's it's a pretty cool tool for that reason. It does have some limitations in that there aren't that many COBRA models available because they take a really long time to develop. So as I had mentioned earlier, sometimes you actually need to develop your own tools in order to work with your own data. All right, but as I said, you can also use Escher to do flux balance analysis. So this is cool for visualizing data. You could put reaction data on here, and so your reaction nodes would be different widths, indicating that they were present or absent in your system. But the flux balance analysis, I think, is one of the more interesting aspects of Escher. Okay, so we'll go back to the main menu here. And then right on the main page here, you can access the FBA. So we'll launch that. And I'm just going to go over a few examples that were in the manuscript, which will be uploaded to your tutorial data set. So this is an example of E. coli. The objective function here is to generate biomass. So that is this function in this lower corner right here. So we have quite a few different reactions that are going into that function. As we mentioned earlier in the lectures, the objective function is what you're trying to maximize in your system. Okay, and that's also shown here in this corner. So we'll be keeping an eye on that throughout these different things that I do to the system. So one of the things you might want to do with a flux balance analysis is look at different sources of carbon for your system. That's probably one of the more interesting things you can do in biochemistry, in bacterial biochemistry at least, see what your organisms are actually eating. So in general, E. coli likes to eat glucose. We can see that here, it's intaking glucose through this exchange reaction. So in this case, when the metabolite is going into the cell, it's actually a negative flux, indicating it's coming from the outside going into the cell. And then if there is a flux of 0 or a positive number, that's going out of the cell or not being removed from the cell at all. So in this case, let's take a look at exchanging glucose for succinate. So succinate is now our new source of carbon. So right here is our succinate exchange reaction. So right now, as I mentioned, if we have a flux of 0, that means it's not going in or out of the cell. So we're going to change that to a -10, indicating it's going into the cell. And we see here that it does go indeed into the cell, and that slightly changes our objective function, which is increasing the biomass. And we did increase the biomass from 0.8 to 1.3. So that's cool. But now what if we take out glucose? So now we have glucose and succinate, but what if we take out the glucose, which is the original source of carbon? So you can do this either through just setting your lower and upper bound to 0 or knocking out that gene. So essentially, again, setting your lower and upper bounds to 0. Okay, so this enzyme no longer works. We're no longer able to actually import glucose into the cell. And so that actually dramatically decreases our objective from 0.8 to 0.4. Okay, so we saw that E. coli prefers glucose to succinate as the source of carbon for increasing biomass. All right, now let's look at a different potential condition. What if we're looking at anaerobic conditions? So no oxygen is in the environment. Right here, we have our oxygen exchange reaction. So if we set this to 0 by knocking it out, shoot, the cell dies. So that means that E. coli cannot survive in anaerobic conditions if succinate is the only source of glucose, or source of carbon, not the source of glucose. So let's reset this. That didn't work. The cells will die, so we don't want to do that. So we're going to reset. So now we have glucose as our main source of carbon. Succinate is not the source of carbon. We're again going to knock out the oxygen exchange reaction. And now the cell survives, though we do have a lower biomass flux. So in general, E. coli would prefer aerobic to anaerobic conditions. All right, so one last example of how you can use this tool for flux balance analysis is determining the maximum yields of different precursors and cofactors. So one of the most important cofactors in any cell is ATP. So we're going to reset this. And this time we're going to set our objective to be ATP maintenance. So that's this reaction here, which is converting ATP to ADP. All right, so we're going to now click the compound objectives because we want to change our objective from biomass to ATP maintenance. We're going to then go hover over this enzyme and we're going to maximize it. All right, so when we maximize ATP maintenance, now we get the flux of 175 and the units are millimoles per gram per hour. So that's not too important, but we do see a much increased flux of ATP. And then we also see that the ATP is being produced through glycolysis here, as well as the TCA cycle. Okay, now what if we wanted to look at ATP maintenance when succinate was their source of carbon and not glucose? So again, we're going to keep this function maximized, and we're going to input succinate into the cell. And we're going to knock out the glucose gene. All right, so now no glycolysis is occurring. Only ATP is being produced through the TCA cycle. And we do see a decreased ATP flux. So seen here, got 82.5 as opposed to 175. So definitely more ATP is produced when glucose is the source of carbon rather than succinate. All right, so that's just a really quick introduction to doing flux balance analysis using new modern tools. So you don't have to go in an Excel spreadsheet and do it all by hand now. You can use these cool tools. They do have some limitations, obviously. They don't have models available for every organism. And mostly they just have the main organisms that are studied by many researchers like E. coli or Homo sapiens and stuff like that. But I think in general, it's going to make flux balance analysis much more practical, and people are going to be able to share really cool data sets with each other. So I hope you enjoyed listening to this short tutorial, and I hope this helps you to understand flux balance analysis a little bit better. And I do encourage you to go out and try it. It's actually pretty fun. It's not that hard to use. I'm not the best with computers, so if I can do it, that means you can too. All right, have a great day and good luck in the rest of the course.